One day I woke up and I asked myself, how can we improve CAR T-cell therapy using precision gene editing techniques? I mean, isn't that what everyone thinks about when they wake up in the morning? I think there's a person there. But seriously, CAR T-cell therapy is one of the most promising breakthroughs in modern medicine. And today, I'm going to dive into how we can use precision gene editing techniques like CRISPR to take this a step further. In this video, we'll go into my review paper. Pause. I've linked my paper in the description box below, so you should definitely go check that out. Play. On CAR T-cell therapy, exploring how it works, what it does, and why precision gene editing techniques are important for its future. CAR T-cell therapy, like any advancement, has its own hurdles, and we'll also be going over that too. Let me break it down for you. To understand how CAR T-cell therapy works, we first need to know what T-cells are. If you haven't watched my previous video on improving the efficiency of T-cells, I'll link it up here. T-cells are white blood cells that play a crucial role in our immune system. They're like the detectives and defenders of our body, always on the lookout for threats like infections or cancerous cells. Each T-cell has a receptor that detects a specific antigen, allowing them to target and eliminate these dangers. There are two main types of T-cell receptors, or TCRs. Alpha-beta TCRs, which make up 95% of T-cells and detect specific antigens presented by MHC molecules, and gamma-delta TCRs, which only make up 5% of T-cells but can recognize antigens directly without MHC molecules, offering broader protection. Understanding these receptors is key to designing CAR T-cells because they tell us what the T-cell targets and attacks. T-cells can also be divided into two main subsets: CD4 plus helper T-cells, which are the coordinators activating other immune cells to help join the fight against cancer, and CD8 plus cytotoxic T-cells, which are the soldiers directly killing infected or cancerous cells. CAR T-cell therapy involves modifying a patient's T-cells to express a synthetic chimeric antigen receptor, or CAR. So far, CAR T-cell therapy has been groundbreaking in treating blood cancers and hematological malignancies like lymphoma and leukemia. A CAR consists of three main parts: an extracellular domain to recognize the antigen, a transmembrane domain to anchor the CAR to the T-cell, and an intracellular signaling domain to activate the T-cell once the antigen is detected. The CAR's design is crucial to ensure that the T-cell can locate and destroy the cancer cells effectively. This is where precision gene editing techniques like CRISPR-Cas9 come in. They allow scientists to make highly targeted modifications in T-cells, enhancing their ability to recognize and attack tumors. And the best part? CRISPR can help minimize off-target effects, making the therapy safer. Researchers have discovered that deleting immune checkpoints like PD-1 and CTLA-4 can significantly boost the persistence and activity of CAR T-cells, leading to better therapeutic outcomes. In clinical trials, combining engineered CAR T-cells with PD-1 inhibitors has shown dramatic improvements in T-cell activity, tumor progression inhibition, and even median survival rates. Gene editing technologies can also optimize immune synapse formation, improve synapse stability, and knock out inhibitory molecules. These modifications can lead to more effective anti-tumor responses by strengthening CAR T-cell activation and enhancing their cytotoxic activity. CAR T-cell therapy has shown great promise in treating cancers, but challenges like cytokine release syndrome, or CRS, neurotoxicity, and limited efficacy in treating solid tumors still remain. Think of CRS and neurotoxicity as fire alarms. They're supposed to signal danger, but when they're too loud, they cause chaos. And solid tumors are harder to treat due to physical barriers and immune suppression, which is why they haven't showed many results in clinical trials. Another challenge is the tumor microenvironment. To help T-cells thrive, scientists are engineering CAR T-cells to resist these suppressive signals that the tumor sends out. Enhancing T-cell persistence is also key, and genetic modifications can help them survive and stay active for longer in the tumor. As of 2023, CAR T-cell therapies have been FDA approved for hematologic cancers, showing remarkable success in treating leukemia and lymphoma, with promising long-term outcomes for children with relapsed acute lymphocytic leukemia. Advancements in tumor-infiltrating lymphocyte or TIL therapy, T-cell receptor or TCR therapies, and off-the-shelf CAR T-cells are expanding treatment possibilities for solid tumors as well. Researchers are improving CAR T-cell therapy by enhancing efficacy for solid tumors, addressing safety concerns with safer gene editing, and expanding its use for autoimmune diseases, infections, and cardiovascular conditions. FDA-approved therapies like Abecma and Carvykti show promise for treating multiple myeloma, and advancements like off-the-shelf CAR T-cells and multi-antigen targeting are helping address current challenges. The future of CAR T-cell therapy is bright, and with advancements using gene editing and other techniques, we move closer to a world where cancer is no longer a formidable adversary. Thank you for watching!